Do you know your kidneys filter around 1100 to 1200 ml of blood a minute, and this makes around 1 by 5th of the blood that is pumped by each ventricle of your heart in a minute. Now, let's check out where actually this filtration happens and how this filtration actually takes place. This filtration happens in the filtration units in your kidneys. The basic filtration units in your kidneys are nephrons, and there are around a million of these nephrons in each kidney. These nephrons, the very first part of these nephrons is what we call glomerulus, which is essentially an intricate a mesh of capillaries. Now see, for this reason, we also refer this ultrafiltration that takes place in your kidneys, that takes place in your nephrons. We also call it glomerular filtration. Glomerular filtration. Let's first check out how this glomerulus looks like and how filtration takes place in it. If you check out this artery here, it's actually a branch of renal artery, and this is what we call efferent arteriole. Okay, this efferent arteriole, which is a branch of renal artery itself is a branch of dorsal aorta, so it brings oxygenated blood into this bunch of capillaries, into this mesh of capillaries. Okay, this mesh of capillaries is formed by your efferent arteriole. Okay, this mesh of capillaries that you find here, okay, this mesh of capillaries, this is what we call glomerulus. Okay, this capillary, this mesh of capillaries. There is another arteriole that actually leaves this mesh of capillaries. We refer to that as efferent arteriole. Now see, and this part which actually surrounds your capillaries, let me draw it with a different color. Okay, this part that you can see over here, which actually this tubule which surrounds your glomerulus. Okay, this tubule over here, this is called, this is named after a British scientist, Bowman's capsule. So you call it Bowman's capsule, or we also call it a renal capsule. Okay, and the space within this Bowman's capsule is the renal space, or you can call it Bowman's space. Okay, this is the space. Now see, when blood enters into this glomerulus, okay, so when it enters into this glomerulus, there are a lot of substances in it which actually leak out. They leak out into this space. For example, the water in the blood leaks out. Okay, electrolytes, Na positive, K positive, bicarbonate ions, okay, they leak out into this space. H positive, they leak out into this space. There are certain dissolved substances like glucose, it leaks out, amino acids are there, urea is there, okay, some creatinine is there, so they all leak out into this space. Now see, when it leaks out into the space, there are actually three different membranes through which it leaks out. Okay, so there are three membranes which actually make the filtration barrier. Okay, so if we check out, let's have a closer look here. If we just take a closer look at this, let me erase this to make space for this. Okay, let's have a closer look at this. Okay, let's check out if this is the blood capillary. Okay, let us suppose if it is this blood capillary. Okay, and this is the capillary wall. We know that the wall of the capillaries is lined by a single layer of cells, and this single layer of cells is referred as endothelium, or these cells are referred as endothelial cells. Okay, these are endothelial cells. So this is the endothelium of the capillary wall. Now see, the blood that enters into this capillary, it contains a lot of components. Okay, it has the formed elements like the red blood cells, it has white blood cells. Okay, there are electrolytes in it like Na positive, K positive, H positive. Okay, bicarbonate ions, there is glucose in it. Okay, there is urea in it. Okay, there is a lot of water which makes up most of your blood, so there is water in it. So most of these substances leak out into this space. Now see, this endothelium, which makes the lining of your blood vessels including your capillaries, there are small pores in it, so it is perforated. There are small pores in it. It's perforated. There are pores in it, or we also use the term, this wall is fenestrated. Fenestrated means it is perforated. There are pores in it, and these pores have a diameter of around 10 nanometer. So this is fenestrated. So things leak out through these pores, and even proteins can leak out through these pores. All the proteins are uh, macromolecules, they're large molecules, but they can also leak out through this. Now see, there is another layer that actually surrounds your blood vessels. Okay, which is a fibrous layer containing a mesh of fibers, and these fibers actually provide tensile strength to the walls of your blood vessels as well. This is referred as basement membrane. So this layer over here is the basement membrane. It does contain collagen fibers. Okay, now see, if proteins do leak out through these pores which are there in the endothelium, but this mesh over here, the basement membrane, doesn't it allow, okay, to pass through. So they cannot make it. If they can make it through these fenestrations, these pores, okay, in the endothelium, but they don't make it through this basement membrane, so you won't find any proteins in the filtrate that is leaking out. Then after this, you have this tubular layer, okay, of this Bowman's capsule. Okay, this is the third layer, which is actually an epithelial tissue. So let's draw it this way. So this is the third layer. The cells are very long, like this. These are the tubular cells, and these tubular cells are actually epithelial cells. Okay, so this is actually tubular cells which form the lining of the Bowman's capsule or the renal capsule. These cells are essentially noted down. These are squamous, simple squamous epithelial cells. So this is simple squamous epithelium. So this is simple squamous epithelium. Now see, some of these cells of this layer, they actually go as far as your endothelium and form these foot-like projections. Okay, they essentially hug your epithelium, endothelium, sorry, uh, and hug it and make these foot-like processes. So you can call these processes foot processes. So these are foot-like processes. These are like the digits of your feet, foot-like processes. For this reason, we refer to these cells as podocytes. So they are referred as podocytes. Okay, so pod actually meaning foot. So pod means foot. Okay, you might have heard about pseudopodia, again over there, false feet. So poda actually means the foot, or podia means the feet. So but in between these cells, okay, these podocytes, okay, there are small slits through which the filtrate actually leaks out into this space. So this space is essentially your renal space or the Bowman's space. Okay, so these slits here, these are called filtration slits. So these are these filtration slits. Okay, now see, or we also refer to them as slit pores. So slit pores are filtration slits. The filtrate leaks out through these pores and comes down into this space, the space that you call. So electrolytes, for example, Na positive, K positive, will leak out into the space. Okay, glucose will leak out into the space. These water molecules will leak out into the space. Okay, urea will leak out into this space. Urea is again a micromolecule. So most of these micromolecules will leak out into the space. Okay, and whatever filtrate comes into this space, we call this filtrate GF, glomerular fil filtrate. So this is referred as glomerular filtrate. Okay, and this filtration is what you call glomerular filtration. Okay, so this actually makes the endothelium, okay, the endothelium, the basement membrane, and this epithelium of the renal capsule, okay, epithelium of the Bowman's capsule. Okay, so this is what makes your filter paper, okay, through which the filtration takes place. So these are the three membranes which actually make your filter paper. These filtration slits or these slit pores, they have, do note it down, they have a diameter of around 25 nanometer. And I told you earlier, these pores in the endothelium, they have a diameter of around 10 nanometer. 1 nanometer is equal to 10 raised to power minus 9 meter. Okay, so this, the filtrate that comes over here in this renal space or Bowman space is what we call glomerular filtrate. Now the amount of filtrate that is formed, the amount of filtrate formed by your kidneys amount of filtrate formed by kidneys in 1 minute. Okay, we call it GFR, glomerular filtration rate. Okay, so GFR refers to glomerular filtration rate. This is referred as glomerular filtration rate. And for a normal healthy person, this GFR is around 125 ml per minute. Okay, so 125 ml per minute. If you check out how much of glomerular filtrate is formed in 1 hour, so 125 multiplied by 60, there are 60 minutes in 1 hour. And if you check out for a day, do multiply it by 24 hours. Okay, and you will see 125 into 60 into 24, it will be 1,80,000 milliliters. Okay, if you can convert it into liters, so divided by 1000. Okay, so that will be 180 liters per day. So your kidneys make around 180 liters of glomerular filtrate each day. Okay, so if you check out the composition of this glomerular filtrate, it has the same composition as your blood plasma, only the proteins don't leak out into the renal space. Okay, they are sent back by your basement membrane. And one more thing that you need to remember over here is that of all these three layers, okay, uh, the first one was the endothelium, the second is the basement membrane, and the third is the epithelium of the renal capsule. Of all these three layers, the basement membrane is the main filtration barrier. Okay, do remember this down. So basement membrane is the main filtration barrier. Okay, basement membrane. Okay, that is the, you can check it out here as well. Okay, so this basement membrane here, this basement membrane. Okay, this is the main filtration barrier. Okay, so this is the main filtration barrier. So this filtrate formed now, then it will go through different parts of your renal tubule, and most of it will be reabsorbed. Thank you.